So yeah, we are. There's going to be a little bit of redundancies between what was discussed before and what we're going to talk now, but it's going to be more focused on terrestrial vertebrates, monitoring for terrestrial vertebrates. So I just want to start with this classical limitation that we have with the traditional methods. Some of them, so they are good for direct observation when you have abundant species. When you cannot have direct observation, you still need to have signs that are abundant so you can observe them. It requires also trained observers, and that also leads to like some pretty frequent misidentification of signs. So this problem of positive, false positive, false negative is also very frequent in traditional methods. So survey of rare and elusive species are also sometimes expensive due to technology required or the field efforts to collect a good amount of data of good quality to make good estimates. Also, individual-based monitoring is often challenging and sometimes impossible when you cannot identify the animal through that natural markings or tag. So what can actually eDNA do? So we were very interested in this aquatic, but also terrestrial biodiversity inventories, trying to see what we can really do with the information that is collected. We've seen that some mammals were very often picked in the water. So that seems to be a way to go, but we really wanted to know what was the best method, not only where to go, but what kind of samples to take to optimize the chance of detecting these animals. Also, a question that was asked was the possibility to identify a species from its track, but also the individual from the track. And a very important question is the cost efficiency of eDNA versus traditional methods. So samples, I'm not gonna talk, it's like the different kind of samples that you can collect are all considered like environmental DNA samples. So I'm gonna talk a little bit more now about the individual identification that we came across at the beginning of our interest in eDNA. One of the questions was to try to see if there was a better, more efficient way of identifying polar bears, because the traditional methods requires the polar bear to be darted, sedated, and marked with some samples collected, and then, so it's very, very expensive. So we did some tests at that time. I'm just showing here. So classical genotyping, when the hypothesis was that it would be possible from the snow underneath the footprint to retrieve some DNA that the bear had shed when he was walking on the snow. So, but we expected this DNA to be very rare. So we tested some next generation sequencing methods to be able to cope with that. So that's what we did. So we expected to collect one footprint, run one analysis, and identify one individual. So what we obtained, we did that on a few bear samples, and we did get a genotype, but that was only from artificial, well, not completely artificial, but the paw print was made with a dead bear paw, and that's the only one that allowed us to do the full genotyping of the bear using this snow sample. We were able to retrieve, although mitochondrial DNA from the snow footprint and from one that was collected in the wild. So there's still some work to do here, but there are some future research we hope will lead to the possibility of doing this genotyping from a more abundant, from a very abundant source of information, which is the footprint. So we were also interested in looking at the possibility to do that in a tropical environment. So we went to the sanctuary of Sumatran rhino, and we did collect this time not from snow sample, but from the mud. So we swabbed the mud inside the footprints of Sumatran rhinos that we kept in a buffer. And from these samples, we extracted the DNA, mitochondrial DNA, and checked if that would lead to the identification, at least, of the species. Because in this place, we have a problem, of confusion between rhino sign and tapir sign. So we really wanted to get a method that would allow us to distinguish these two species from their footprint. So, and the results showed that 30% of the sample, of the footprint that we sampled, showed enough DNA that allowed us to identify the species, to confirm that the DNA from a rhino. So that was a very positive outcome, and again, with in the future the possibility to collect nuclear DNA to genotype the animal from its footprint. So, and I'm gonna talk now about biodiversity, and specifically terrestrial biodiversity, because what we've seen is that, yeah, it's the studies that have been conducted show that eDNA really allows to inventory pretty well those aquatic species, but the situation for terrestrial species seems more complicated. So while I'm just going through this, it's just the metabarcoding and how it works. I'm pretty sure you have understood that from the beginning. So the two questions that we had were, the first, the ability to detect rare species from water sample. Like really with the assumption that water, like it was said a little bit before, really collects information from an entire watershed. So rather than going and walking through the entire environment, can the water collect this information on our behalf? And then by just collecting this water, is it enough to detect a rare species? So that had a really, really important application for the detection of Sumatran rhino in West and Central Kalimantan. There is a huge habitat that has never been surveyed yet, and that could really benefit from this new technology. So we just made a very quick test and went out, collected a few eDNA samples from the rivers in an area where the rhino was present. At this time, we did collect only 10 liters of water, and the results that we got was lots of terrestrial species as in previous studies, lots of fish as well, amphibians, but no rhino. So, and like few small cats as well. So the question that we have now is like really try to answer the question of what does really mean the information that we collect in this water, and is eDNA a good method that we could use for instance, monitor tiger and tiger prey population and detect very rare terrestrial species. So one of the problem with eDNA from water is that most studies are focused on detection and not really on abundance. Few studies have compared this eDNA abundance with estimates of relative abundance, and it has been applied only to aquatic species. So for eDNA and terrestrial vertebrates, it has remained largely unresolved. And the study that we are presenting in, I'm giving it over to Robin for introducing our study, is to try to understand better this situation and answer these questions. Over to you, Robin.
Thanks, Arnaud. Yeah, so this kind of gets back to a point that Kat raised in the first presentation, where she was mentioning looking at these concentrations of eDNA for the various species that you find in water samples, and how does that relate to the abundance or density of fish species from electrofishing? So, I believe she mentioned that there's good correspondence in aquatic systems by and large. As Arnaud just mentioned, in terrestrial systems, this is really unknown. So the focus of this project that I'll talk about now is really to compare wildlife abundance from camera traps and eDNA in the South Chilcotin Mountains. So camera traps, as we probably know, are a very standardized, well understood way to measure the relative abundance and in some cases the densities of a variety of wildlife species that are well surveyed by cameras. So we aimed basically to look at this question in this particular area of British Columbia. Arnaud, you can go to the next slide, please. Thanks. So hard to see this map here for anyone who knows the west coast of Canada. This study site is about 4 hours north of Vancouver. And what we did was lay out a grid of camera trap locations. There's roughly 60 of them that covers about 800 square kilometers or so, laid out in the way that we see here. So that area in green is a provincial park. Half the cameras were in that park, half were outside the park, largely in forestry areas. It's not showing up on this map here, but there's also quite an array of streams and rivers throughout this area. So it presented a nice opportunity to look at whether the eDNA that does wash into these areas carries enough signal for terrestrial wildlife so that they can be not only detected, but also potentially measured in terms of the concentration and abundance of eDNA for those different species in the different samples, and to see how those compare from estimates generated from a more sort of standardized way of monitoring terrestrial wildlife via camera traps. Next slide, Arnaud. So these are some of the images that we've generated from the camera trap survey. So it's an area where there's a fairly intact large wildlife assemblage. And so, analogous to some of the sites in the tropics where we work, where there's still, the sort of largely full complement, of medium to large wildlife species that you might find in a system. Then go on, Arnaud. Next slide. Yeah. So it's also, a system that nevertheless there's humans present throughout the system. And so, one of the nice things about camera traps is that you can get measures of both wildlife species and also human activities. And so, the same data, same method is generating both of those. Obviously for eDNA, you can pick up humans, but you can't pick up necessarily what you're doing. So that's one of the differences from this method versus the other. Next slide, Arnaud. So basic design, the data that I'll present here is from about 60 camera traps. As I mentioned, 800 square kilometers roughly that it's covering. They're about 3 kilometers apart. These data are just from a few months in the summer from 2018, but we've now also got data from this summer that's coming in. And I think Arnaud will present the eDNA analog to the camera trapping also from 2018, but there's more data that's coming. Arnaud, next. So this gives you an idea. This is a less sophisticated way of presenting simply sort of relative abundance of the different species that we detected from camera traps. So you can see that this is the top 10 list of species of wildlife that we detected from cameras. Next slide, Arnaud. But then if you add in the humans to the system, we can see that, despite the fact that this is, really a wilderness area, humans are actually dominating the detection rates at this site. One of the things that was surprising though, next slide, Arnaud, was the array of different species that we detected. So even though these cameras are largely geared for medium to large sized terrestrial wildlife, we actually found a huge number of birds on the camera trap. So 33 bird species, as well as the 28 mammal species that we detected. The table basically here lists the number of detections. And so again, the key question that we wanted to look at were the number of detections of these wildlife species that we found via cameras related in any way to the abundance or concentration of eDNA in the water samples. So I'm going to turn it back over to Arnaud now to let you know what we found on that front.
Okay, so yeah, this is the kind of place where we went to do the sampling. So we used a vampire sampler and a filter, and we filtered for 30 minutes water from the river. And we did that over the entire area where we had set up camera traps. So we tried to match every camera trap or river basin that was covered by camera trap with at least one sample of water. So that was a pretty huge effort to go and collect all this information. We were also very interested in trying to look at the optimal river size for collection of the different group of animal. So this is one of the main results that we have. So it shows the taxa that we have detected in the mammal, for the mammals. So we detected 21 taxa. The result doesn't really show all the species because of the problem or the limitation of the reference database. Some species that were possible to identify via camera traps were not identified with the DNA. But it shows the different species and the most abundant one, which are here the small small mammals. It seems to be really good at picking small mammals. Mule deer were also pretty good, and some species that have some more aquatic behavior, of course, were more frequently detected, like the castor or muskrats. We also detected, in addition to that, 7 fish species, 41 birds, and 2 amphibians. I'm just putting the same kind of graph showing the frequency of the detection of the same taxa on the camera trap, and we can see that the distribution is pretty different. The camera trap seemed to capture much better cat species than eDNA did. We detected very few cat species. So we were also interested in what we mentioned earlier, in trying to understand how the quantity of DNA that we would detect in the water was related to the abundance or relative abundance of the species in the watershed. So we selected some samples that were collected in the main river, and so compared the data obtained, the quantity of DNA from these few samples and the frequency of detection of the species with the camera trap in these 2 watersheds that are represented here on this slide. And we plot the results only for, like, we grouped the different species together for the mustelids, for instance, for the cats, and I cannot see what is on the right, and the deer. And so showed that so that's log-log relationship between the quantity of the number of copies of DNA that we found in these samples and the total number of pictures that were collected on the camera trap during the entire season. So it shows that there is some kind of relationship but it's still not really clear. We have to replicate, and this is one of the objectives of the study that we are continuing there. Now we are focusing on trying to collect even more water from the same location I have showed previously, to see how this relationship changes over time, if it's frequent or if we just were very lucky and we have a pretty good relationship. So this is basically what the next step that we are gonna do before transferring this technique to the study of tiger and tiger prey in Southeast Asia. So one of the promises for eDNA is clearly its potential for non-invasive sampling of biodiversity. It's a new technology that is still in development, but I really believe that it can revolutionize our understanding of biodiversity, where it's found, how abundant it is, and be also a much more cost-efficient technique for this kind of study. And it's also, with all the research that is done at the moment, it's very likely to improve in the coming years, both in its efficiency and in its cost. Thank you. Over.
Wonderful, thanks guys. We'll have some specific questions for Arnaud and Robin, if you want to drop them in the chat. You mentioned cost. There was a question that came up earlier about cost per sample, and you mentioned cost effectiveness as a part of this, the value of eDNA and this technique. Do you have can you answer the question about how much it costs versus other sampling methodology?
I think the answer can be pretty different, like depending on the question you're asking and what you're looking at, what you're including in your costs. But so if you just compare the costs, for instance, of what we're doing for tiger detection, like just the occupancy survey, so going and relying on the detection of the signs to be able to assess the unbiased presence of the species in a particular area. Usually it's a 15 or 16 by 16 square kilometer. People need to walk between 40 and 100 kilometers to get good enough detection probability of the species when it's present. So the collection is on tiger and other mammal species, and that costs, I think, I would say between $5,000 and $10,000. So if you really if the sampling of water in a river can really be can demonstrate its accuracy and efficiency, and if you need just one sample or two samples to cover the same area, then you could probably do the same sampling for less than $1,000.
Wow, that's crazy.
So cutting the cost by 5 or 10.
Do you know how much the cost is per sample?
It might be different, but it's around like $300–400.
Okay, cool.